Good afternoon. In today's practical, I'm going to talk about some of the methods which we use to isolate and diagnose viruses in the lab. As we mentioned in the lecture, there are different types of viruses, and those tend to give us different infections. For that purpose, we mentioned that the viruses vary in their sizes and their structure, and hence we ought to find some ways of detecting these viruses so that we can handle them and manage them and also treat them in the appropriate way. Therefore, the learning objective of this lecture is to understand some of the principles of those diagnostic methods and understand why the reason why we need to use them, and some applications. Also list what are the current tests available in a diagnostic virology lab. So until a few years back, virology diagnosis was outside the mainstream of clinical laboratories. And the reason why is mainly because the virology laboratories a few years back depended on some simple methodologies to detect, and such as tissue culture techniques. Those techniques were really slow and expensive. The availability of the commercial reagents was very limited, and also few treatments were available during that time. With time, things started to change, and we saw that diagnostic methods also started to develop in a different direction. So now I'm going to mention those diagnostic methods and how they developed really to help us in detecting those viruses. Now, for example, earlier we had something called the direct examination. Now, direct examination, what does that mean? Clearly, it means that you directly look into a specimen to detect the presence of the virus. This can really be achieved by different methods or ways, either by the use of a light microscope. And what we tend to look here is we tend to look at certain histological changes that can give us a clue that there is an infection, and certain bodies that are left over of the process of virus replication. The other method is by the use of electron microscopy. And here we tend to look for the morphology of the virus particles. And this can also be modified a little bit to be used with immunology. So we refer to that as immune electron microscopy. And among the third direct method of examination is the antigen detection, which we tend to use different ways, such as immunofluorescence, ELISA, and we will come to talk about them as we go on. With direct examination, as I said earlier, we tend to look for certain histological appearances or changes, and this is achieved by placing a specimen on the stage of a microscope, and usually we look at what sort of changes happening in comparison to the normal piece of tissue, for example. And we tend also to use certain stains to detect those left over of the process of replication of the virus, or what we refer to as the inclusion bodies, to really help us in diagnosing the presence or absence of those viruses. Among the direct examination methods is the use of electron microscopy. Electron microscopes were discovered or were invented at the early years of the 20th century. They became very important tools for detecting very small, minute particles, such as the virus particles. The principle behind the electron microscope is to place a specimen within the electron beam, which is directed from a cathode to the anode, and this beam goes through the specimen. What we tend to see here is a negative image or a deflection of the image of the virus particle. And usually this is in black and white. So there is no color, there is no immunofluorescence, nothing can be seen here except for a negative image of what is being captured through the deflection of the electrons from the surface of the specimen. For that purpose, electron microscopes really are very good instruments to detect virus particles. And however, we require something like 10 to the 6 virus particles per ml to be able to visualize these virus particles. Usually the magnification is quite high, something like 50,000 times the normal size, meaning that if we have 1,000 times magnification in the light microscope, here we can magnify it up to 50 times more. Viruses can be detected in different types of specimen for that purpose. For example, it all depends on where the viruses are infecting. Feces can be utilized, vesical fluids, skin scrapings. All these can be used in different ways and preparations to detect certain specific viruses. As you can see here in this electron micrograph, there are certain viruses which are unique in their structure. And for example, in adenovirus, you tend to see those fibroblasts which are protruding from the virus particle. And this is very unique to adenovirus. In comparison, rotavirus is a little bit different. And usually what you tend to see here is a wheel-shaped structure. And hence the name rota. Rota means wheel spikes. And therefore, the structure of rotavirus is a little bit different from adenovirus. However, generally speaking, most viruses or human viruses are spherical in structure. Hence, this really makes it difficult really to distinguish between one virus particle and another based on their appearance or the structure. For that purpose, people try to develop different ways and methods to isolate viruses and identify them. Among those methods is what we refer to as the indirect examination. And here, what we mean by indirect is we try to isolate the virus and amplify that. Amplify that to the extent where we can use certain methodologies to detect. Among the indirect examination is the use of animals. Now, we said earlier, if we want to know whether a specimen has a certain number of viruses to be detected, it has to be something like 10 to the 6 virus particles. However, certain specimens may contain far less virus particles. Hence, at the beginning, what scientists did is they used to take animals and take a specimen from a sick person, inject them into those animals, and try to see what sort of effect this is going to cause towards that animal, whether it's going to cause a disease or it's going to kill the animal. And usually what they will do after that, they will dissect the animal and look for those changes which have occurred in certain organs. However, this is a very really primitive way of looking into things. It's not that useful because you require quite a lot of animals, and sometimes these results could be fruitless. So somebody thought, why don't we come and use eggs? And the idea came of injecting eggs with specimen of viruses and leaving them in an incubator for a certain period of time for that the viruses can replicate and then try to detect the presence through looking at the changes which has occurred within the egg. Of course, what we tend to see here is we see the appearance of certain pox-like structures, and even inclusion bodies can be identified. And for that purpose, you ought to really take that further on by removing those pox and trying to identify the virus by hemagglutination test or inclusion bodies and so on. Again, the use of eggs really meant that it will take quite a long time for us to identify the virus which we want to isolate from a patient, and moreover it takes quite a large number of eggs for that purpose. For if we have 200-300 patients, that means that we have to keep on infecting those eggs and keeping them in a good condition and so on. So this is really sometimes not that practical in these terms. So the idea came with some scientists is, why don't we use cells? And the cells which we take from certain organs of the animals can be utilized to grow those viruses. And what we can see here within those cells is what happens through the process of infection in terms of the cytopathic effect. This can be tested by different ways, hemabsorption, immunofluorescence, and so on. For that purpose, cell cultures became very widely used for amplifying viruses and isolating them. We know that there are 3 types of cell cultures. There is what we call the primary cells, for example, monkey kidney cells, meaning that those are cells which are taken from a monkey. And usually you take those cells, you smash them, then you tend to put some nutrient, vitamins, media, and you put them in a flask and you grow them. Then there is the other type of cells which we refer to as the semi-continuous cells. Usually those are human embryonic kidney or skin fibroblasts. And those, as I said, are obtained from embryos, and they are utilized for the purpose of growing viruses, simply because those cell types, the good thing about them is they can be passaged. In primary cells, usually those cells, if you take them from the petri dish after growing, you might be able to grow them again once. However, if you try to do that a second time, you might not be able to grow them anymore. With semi-continuous cell lines, no, the situation is a bit different, where you can really passage them several times, sometimes up to 40 times you can passage them. And this is really good in a sense that you can store these cells in the freezer, and whenever you need, you can take a batch of those to infect with a specimen. Finally, we have what we know, what we call the continuous cell lines, and the continuous cell lines here are vary. Some of them are referred to as HeLa, Vero, and Hep2, and so on. They vary from their origin, but mostly those are cells which are taken from tumors, cancer cells. And the reason why we call them continuous, because whenever we want to grow them, we can passage them so many times. And in theory, we can passage them up to infinity, simply because, as I said, those are tumor cells or cancer cells that have the capability of growing infinity to infinity. As I mentioned earlier, primary cell culture are widely acknowledged as the best cell culture system available. They support the widest range of viruses. However, they are very expensive and difficult to obtain, simply because we said primary cells are taken directly from the organ, meaning that we have to bring the animal and take the organ and so on. And for that purpose, we need so many animals, which are not available all the time. Continuous cell lines are easy to handle, and there is one issue, is that the range of viruses supported here is often limited to certain types of viruses. So that's what makes those cells really more specific for the use of or amplifying certain types of viruses, while the semi-continuous are the best probably cells that are utilized for that purpose. As you can see here in this diagram, we just wanted to show you that, for example, mice tissue are taken from mice, then digested with different types of material, cells washed, then left in a petri dish and left to grow. And from there, they are transferred to a flask with the addition of different nutrients. They will be left to grow for a certain period of time until they become or they form a monolayer on the surface of this flask. As you can see, usually cell culture methodologies or techniques are used in very sterile conditions. So the lab should be very sterile, simply because you do not want to contaminate the cell lines with any bacteria or any virus. So you have to have a very clean environment for that purpose. As I said, once the cells grow and reach or form a monolayer, you can really detect them or look at them by the use of what we call the inverted microscope. Now, this is a little bit different from the known microscope, where the light usually comes from top, while with the normal microscope, the light usually comes from below the specimen. So that's what makes those type of microscopes, or what we refer to them as inverted microscopes, unique and important for the use of tissue culture. Now, when we grow cells and they become confluent, we usually infect them with a specimen of viruses. What do we expect to see? once they are left in an incubator for a certain period of time, meaning that the cells are infected with the virus for a certain period of time. What we aim for is to detect what happens really within those cell lines, meaning that we look for the cytopathic effect. And the cytopathic effect usually can be in different formats or forms, either by forming what we call the ballooning of the cells, meaning that the cells become rounded because of the infection, and usually the cells, for example, the fibroblasts, usually they grow on the cell surface and they usually are attached to the cell surface. But when they become infected with the virus, they tend to lose this capability of attachment or adherence. And for that purpose, they start to become rounded, and finally they get detached from the surface of the flask to become floating within the media. Another type of cytopathic effect is what we call sensation formation. And usually what happens here is within the cell lines you will see, or within the cell lawn where the cells have grown, you will see very large, giant cells with multinuclei. And the reason why is because those viruses, when they infect the cell, they do not leave the cell. They infect the cell, next cell, which is attached to the cells that's infected, and the membrane starts to disappear between those cells, and therefore they start to form a very large cell with multinuclei. And this is what I just mentioned is called sensation formation. Now, other than that, sometimes what we tend to do is we tend to really try to detect the presence of viruses through certain techniques, such as what we call heme absorption. And usually this is done by depending on the cells acquiring the ability to stick to mammalian red blood cells. And however, this methodology is not that accurate. Therefore, other better techniques have been utilized and used for the detection of viruses. As you can see here, this is within the cells or cell infected, whereby there is the rounding of the cells in both those specimens. First specimen represents enteroviruses, while the second specimen represents herpes simplex. As you can see, those here sometimes they tend to form what we call sensation formation, a very large cell with multinuclei, and in this case the rounding. Again, this is another picture of RSV, or respiratory syncytial virus, which tends to form these giant cells, multinuclei cells, and as you can see here, some of them have so many cells, and also within measles in certain cases. Now, usually we tend to use cell culture techniques to isolate certain viruses. For example, herpes, cytomegalo, adeno, polio are usually best grown and isolated by cell culture techniques. However, less frequently isolated viruses, for example, measles, rubella, rhinoviruses, Coxsackie, they are less used or less frequently isolated by cell culture techniques, simply because, for example, in rhinovirus, the infection usually lasts for a few days. So it's not really worth to go and grow the virus for the reason of amplifying and detecting what sort of a virus is present. There are, again, rapid diagnostic methods for detection of viral antigens, but the important thing is that you should be able to know what sort of a specimen you ought to take for that purpose. As we said, each virus has its own the specific specimens have to be taken for that purpose. For example, nasopharyngeal aspirates are used for influenza, parainfluenza, adeno, and so on. Skin can be utilized for detection of herpes simplex, varicella zoster, and blood probably used for cytomegalo antigen, and so on. As we mentioned earlier, there are many diagnostic methods in virology. And we talked about direct examination, we talked about indirect examination, where we try to amplify viruses using tissue culture techniques. However, there are other methodologies, for example, serology. Now, serology, again, within serology, there are different ways of detecting, and we are going to talk about three of these methodologies, and we have usually 3 stations to demonstrate different types of tests that we tend to use in the virology lab. For example, the immunofluorescence. Now, if you recall, we infected cells with viruses. However, when you look at the cells infected, you really cannot distinguish cells infected with virus A from cells infected with virus B through the cytopathic effect. It's really very hard. So what we tend to do is we take a specimen of those cells and we place them on a slide. As you can see here, we place them on this slide. And we usually use, because those cells contain the antigen or the virus, we use an antibody, which is tagged with a fluorescein molecule. Now, if the virus is present in the tissue, the antibody is going to react with the antigen, and once the antigen or the antibody-antigen reaction is excited by a UV light, you will see the emission of an indicator which will tell us that the antibody-antigen reaction is present. Otherwise, if it's not present, things will really wash as we prepare the slides after washing them with different buffers and so on. So the slide, which was prepared previously, is taken and placed on the stage of a fluorescent microscope. Now, the fluorescent microscope is a little bit different from the other microscopes, which mainly here depends on UV light source. And this UV light source usually passes through the slide, which contains the stained cells that has the antibody-antigen reaction, which is tagged to a fluorescein molecule. So once the UV light passes by, this will excite the fluorescein and causes the cells to show in a bright color, such as this, what you see in the picture, the green color. So mainly here what we are seeing is the cells showing the fluorescein molecule. Cells. Again, here there is a specimen of cells infected with the virus and another specimen with cells which have no virus in them. That means that this is used as a control. You will see that once we treat those with the antibody, both of them, usually the antibody is not going to find the antigen in slide B, while it's going to find the antigen in slide A. So therefore, the reaction is going to be in A, and there is no reaction in B, which leads to the cells that are infected to become greenish, while those that are not infected, they will stay reddish, indicating that there is no virus in those specimens. And as you can see that here we have a specimen of herpes simplex, and we also have a specimen of rabies that have been treated with an antigen that is detected by immunofluorescence. Among the other serological methods is the use of what we refer to as latex agglutination test. Now, the principle of latex agglutination test is very simple. Usually we have very small particles which we call latex particles. Those latex particles are coated with an antibody. Now, if you have a virus particle, which we refer to as the antigen or specific antigen, this usually will react with the latex particle which has the antibody. Now, what happens here is there are going to be agglutination, because the virus particle is going to react with more than one latex particle, and therefore they will start to agglutinate and they will start to form clumps to indicate the presence of a virus in a solution. Of course, we have controls that we can see whether there is an infection or there is no infection. So there is a positive control and there is a negative control, which will tell us again that this latex test is working as it should be. As you can see here, this is on the left side, the A sample is agglutination of the virus particles, while on the B side there is no agglutination, just a milky solution, which clearly indicates there is no virus here to be detected. As I said, antibody molecules can be bound to each latex bead, and this will increase the potential number exposed of antigen binding sites, and therefore this will lead to this agglutination process. Latex tests can be used for so many purposes. For example, pregnancy, rubella, herpes simplex detection, rotavirus, HIV. Even for coronavirus these days, it is being utilized for such tests. Among the other serological tests is the use of what we refer to as ELISA, or enzyme-linked immunoabsorbent assay. Usually, ELISA is a plate-based assay technique designed for detecting and quantifying substances such as antigens or antibodies. Among the other names is enzyme immunoassay, or EIA. What is the basic principle of ELISA? Usually, the use of an enzyme to detect the binding of antigen antibody. And this enzyme converts a colorless substrate or chromogene to a colored product, therefore clearly suggesting that antigen antibody is bound in this situation. And ELISA can be used to detect either the presence of antigen or antibody in a sample, depending how the design or the test is designed. As I said earlier, there are two main variations on ELISA method. ELISA can be used to detect the presence of antigen that are recognized by antibody, or can be used to test for antibodies that recognizes an antigen. For example, we have a coated well in these 96 plates. Each well is coated with an antigen. These red particles are antigens. When we put the antibody, for example, a serum sample which contains an antibody, if the antibody binds to those antigens, then after washing, we add an enzyme-linked antibody that will react with the original antibody. Therefore, we would have enzyme-linked antibody binds to specific antibody. And after washing and adding a substrate, this substrate that is added will be converted by the enzyme into a colored product. And as you can see here, the colored product is yellowish, and there are different fades of yellow, which can help sometimes in quantifying the amount of antibody or that is present in a serum sample. You can see here in this plate is used for the detection of HIV antibody, meaning that we have a specimen or a sample of serum taken from a person who is infected with HIV and is treated through the method which we just mentioned earlier. Once we tend to see the color changes, this will clearly indicate whether the person has HIV or not. Of course, we tend to use controls. We have negative controls, positive controls, really to help in identifying the appropriate reaction. Finally, the viral genome detection. I'm not really going to talk about this in this lecture, but I hope that the opportunity will come one day and we can talk about different molecular techniques that are utilized to detect viruses. I think I'll stop here. Thank you very much for listening. I hope this presentation is clear. If you have any queries, any questions, kindly don't hesitate to call me or send me a message or any sort of communication asking what sort of queries you have in your mind so that I can really tackle these questions. Thank you very much for listening, and goodbye.